Okay, welcome to Lecture 1 of my mini immunology lecture series. Today we're just going to briefly do an overview of the cells of the immune system as well as the organs of the immune system and talk about some big overarching concepts of immunology. So the first thing to do is to introduce you to the players because we're going to be discussing these throughout this series. The immune cells fall into two categories, the innate category and the acquired category. And this is kind of a big overall theme, one of the big overall themes of the immune system. The innate immune response is ready to go. It is going to respond to all pathogens, and each cell has a particular type of response it can, or effector response it can deploy in response to different types of stimulus. The innate immune response is, as I said before, ready to act, and the cell types fall into a few different categories. You have your granulocytes, which are your eosinophils, your neutrophils, and your basophil. So all of these are called granulocytes, and we'll talk about each of their individual functions in a future discussion. Your other innate immune cells include your mast cell. Your mast cell is famous for the allergic response, but it also is important during parasitic responses and warm or helminth responses. You have two antigen-presenting cells, so you have your dendritic cell, or your DC, and you have your macrophage. And both of these can present antigens, which we'll discuss in a future lecture. The final cell in your innate response is your natural killer cell. And this implies that it's a killer cell, and it does just that. It goes around and kills cells that are infected with bacteria, with viruses, cells that are stressed, cells that need to be removed because they're damaged in some way, UV radiation or any of those. So your natural killer cell has the ability to identify those types of cells and kill them. Now on your acquired side, this is more of your learned response. Your learned response is very specific, and your innate response is very non-specific. So an example of this is that your macrophage can respond to flagella, and it doesn't matter if the flagella is being expressed by salmonella or is being expressed by shigella or vibrio, cholera. Any type of flagella it can respond to. Your acquired immune response has to be very specific. The cell that's going to respond to salmonella is going to be different than the cell that's going to respond to cholera, for example. So they're very specific, whereas the innate response is very nonspecific. There are two main categories of cells in your acquired immune response. The first is your B cell, and the B cell is going to make antibodies. And the B cell can become a plasma cell, but the plasma cell is part of the B cell lineage. The other type of cell is a T cell, and the T cell express a cluster differentiation number 3, or a CD3. And this cluster differentiation 3 cell can become either a CD4 cell or it can become a CD8 cell. The CD8 cell is a cytotoxic T cell, and the CD4 is a helper T cell. So again, similar to the natural killer cell, cytotoxic T cells can kill targeted cells, but they're very specific. A cytotoxic CD8 cell that kills a flu-infected cell cannot kill a cold-infected cell or a measles-infected cell. So they're very specific to the type of infection or antigen stimulus. Now there's one cell type that's called the natural killer T cell, and it's called that because it has some properties of both natural killer cells and of the CD8 T cell. We're not going to spend too much time on these cells because they're very rare. Most of these cells have been identified to target the cytomegalovirus. Cytomegalovirus is a chronic infection that many people in the United States have, and as you get older, a large percentage of your immune system is focused on controlling the cytomegalovirus infection, and that's where you would find an NKT cell. Now, the next thing we need to talk about is immune molecules. We'll go into specific immune molecules throughout the course of these lectures, but this is just a very general overview. So soluble immune molecules are proteins. They are proteins that are encoded by your DNA, and they are generated and released in response to some sort of activation within the immune system. Now, these proteins are in a general category called cytokines. And cytokines help the immune system communicate with themselves. Two very general or very common types of cytokines are your interleukins, and these have an IL and usually a number, so IL-2, IL-4, there's going to be IL-1, there's going to be IL-3, IL-6, etc. So the IL stands for interleukin, and the number generally dictates in what order they were identified. These interleukins are going to be made by specific cells, and their purpose is to sort of tell other cells of the immune system what type of immune response that is needed. For example, an immune response to an intracellular bacteria is very different than an immune response to an extracellular bacteria. And these types of cytokines will help guide the immune response to hopefully the best types to get rid of whatever the infectious agent is. Another general category is your chemokines. And chemokines are a type of cytokine that start with CXCR or CCR, and they usually have a number after them as well. CXCR4, for example, or CCR5. And again, these are released by many different cell types in response to some sort of stimulus. And they're different from interleukins in that the chemokines are really sort of to direct traffic. They're there in order to make sure that your immune cells go to the right location. If you have an infection in the liver, you want your immune cells to be guided to the liver, and chemokines help the cells get guided to the right location. Now, in order to respond to these soluble proteins, the cells have to have receptors, which are insoluble, but the receptors are important molecules of the immune system, and these receptors are expressed on certain cells under certain conditions. For example, we have interleukin 2. Interleukin 2 is a growth factor for T cells. So when a T cell needs to grow, for example, it's been activated because it's specific for influenza and you're infected with influenza, then the interleukin 2 will help that influenza-specific T cell grow. So that influenza-specific T cell is going to express the interleukin 2 receptor so it can respond to the interleukin 2. Your cold T cell will not be able to respond to interleukin 2 because you're not infected with a cold virus, you're infected with the influenza. So the receptors, the upregulation and downregulation of these receptors are important to make sure that the right cell is targeted by these types of cytokines. Okay, so the last thing we want to talk about in this mini lecture are your immune organs. So you have two types of immune organs. You have primary and secondary. Primary is where the immune cells develop, and secondary is where they typically become activated. So your primary immune organs are your bone marrow and your thymus. And your secondary immune organs are where you have activation. They come in two different types that are generalized by whether or not they have a capsule. So you have encapsulated, which are going to be your lymph nodes and your spleen, and your unencapsulated, which are going to be organs such as your Peyer's patches of your gut and your tonsils, for example. So they're basically just aggregates of immune cells that develop into secondary lymphoid structures. So we'll talk a little bit about that when we talk about specifically your lymph node, because you have regions where your B cells go, where your T cells go, where your innate cells go, and when you get an aggregate like that, it's referred to as a secondary lymphoid tissue. Okay, so first let's talk about the bone marrow. In the bone marrow, you have hematopoiesis, And hematopoiesis is basically the development of red and white blood cells. So it all comes from a stem cell, the pluripotent hematopoietic stem cell. Now, within your bone marrow, you have two types of stem cells. The hematopoietic stem cell and you have a stem cell that's stromal. So from this you get your red blood cells, your white blood cells, and your platelets. And from your stromal stem cells you're going to get fat tissue, cartilage, and bone. Now your red marrow is considered your myeloid tissues, which are your red blood cells, white blood cells, and platelets. And your white marrow are your fat, cartilage, and bone. So the other thing that happens with your bone marrow is that you have B cell development. If you'll remember, B cells are a type of your acquired immune system, and it is made through hematopoiesis just like any other white blood cell. But the B cell, before it can be released into your circulation, has to go through maturation and development, and so it stays within the bone marrow to do that. Now, the bone marrow that we're talking about is going to be in the central cavities of axil and long bones. So there's about 80 bones in your axil system. That's your vertebrae column, facial bones, and then your long bones are those bones that are involved in structure and mobility, such as your femur. And so that's where hematopoiesis will be taking place. Your hematopoietic tissue are referred to as islands that are surrounded by adipose or fat tissue. Now the next organ we're going to talk about is the thymus. And the thymus is the site for T cell development. And so the T cell undergoes hematopoiesis in the bone marrow and then gets released from the bone marrow and travels to the thymus so it can become a mature T cell. So the thymus is considered a primary lymphoid organ because it is developing an immature T cell to become a mature T cell. Now the thymus is also part of the endocrine system. It's going to produce a hormone called thymosin. And remember that an endocrine means that it makes the hormone and releases it into the blood to act on distant cells. And so the distant cells that are going to be responsive to thymosin are your released T cells. And so it helps your T cells to continuously be stimulated. And T cells have a half-life of about 30 days. Okay, so your thymus is made up of a medulla, which is this center pink region. And in the medulla, you'll undergo what's called negative selection. And we'll talk about that in a future lecture. And you also have your capsule. So this is encapsulated. You have your cortex. And this is where you undergo positive selection. You have a thymic lobule. And then you have, of course, the interlobular septum, which is here. The corpuscle, or Hassel's corpuscle, the one known function of these structures within the thymus is that they produce a cytokine called TSLP. And TSLP just stands for thymic stromal lymphopoietin. So anytime you hear poietin, you think of maturation. And so the thymic stromal lymphopoietin is helpful in the maturation of T cells. So TSLP will help T cells within the thymus mature, whereas thymosin will be released and will help to maintain the T cell population outside of the thymus. All right, so the next structure is our spleen. Our spleen is considered an encapsulated secondary lymphoid tissue. The spleen has a capsule. It has regions referred to as white pulp, these little circles. And they're called white pulp because that's where the white blood cells are, your T cells, your B cells, your macrophages. It has venous sinuses, which are these blue. Surrounding the white pulp, you're going to have red pulp. And within the red pulp are primarily going to be red blood cells as well as macrophages. This is where dying and dead red blood cells go, and the macrophages will remove them. Remember, macrophages help to maintain homeostasis, and one way to do that is to remove dead and dying red blood cells. You have also the trabeculum. And so within the spleen, these pockets of white pulp which are going to be aggregates of immune cells that will help to respond to any sort of antigen coming in through the blood. And so this is sort of a general diagram of a white pulp. The white pulp is going to be made up of zones. You have an area called the marginal zone. And in this zone, you're going to have macrophages that trap antigens. Beneath that, you're going to have the B cell zone, and this is where B cells will aggregate. You have shown here a germinal center, and the germinal center is where activated B cells are dividing and making antibodies. You have the perifollicular zone, and you have the PALs, your periarterial, lymphoid sheath. So your T cell zones. And you also have a central arterial. Okay, so the white pulp is there to deal with incoming antigens. Your red pulp is there to remove dead and dying red blood cells primarily. Okay, and the next secondary lymphoid tissue is lymph node. This is also an encapsulated tissue, and it also has B cell zones and T cell zones. You have lymph. So we talk a lot about blood circulating through blood vessels. Well, another circulatory system in your body is the lymph. And the lymph is where the white blood cells travel. And they're primarily traveling to go from lymph node to lymph node. You have about 400 lymph nodes in your body, and these cells like to go from lymph node to lymph node, traveling through the lymph. So getting into the lymph node, you have your afferent lymph. And so these would be going in, so your white blood cells will be entering your lymph nodes using the afferent. And to exit, they use the efferent. E for exit. All right, so the other structures that are, again, going to be sort of redundant from other secondary lymphoid tissues is you have a medulla, so that's this center area. And this is primarily where your macrophages will be. You also have some short-lived plasma cells that will be in the medulla. And then you have, of course, your outer cortex. which are your B cell zones. Okay, and then you have your paracortex. So this lighter pink here. And that's going to be your T cell zones. In your T cell zones, you're going to have these structures that are going to be called high endothelial venules, or HEV. And this is where your T and B cells will enter the lymph node, and then they will migrate to their different sections of the lymph node, the paracortex for the T cell or the outer cortex for the B cell. In the middle of the B cell zones, you have germinal centers. And just like in the spleen, these germinal centers are the site where activated B cells are dividing and releasing large amounts of antibody. Apart from that, you have your capsule because it is encapsulated. And you have your vein and artery. Okay, so T cells in the T cell zone, which is the paracortex. B cells in the B cell zone. This is also called the B cell follicle. You might hear that term. It means the same thing. Outer cortex, B cell zone, B cell follicle is where the B cells are in the lymph node. And then the germinal center is a dark, dense center within the B cell follicle where the cells are actively replicating and making antibodies. And then our last organ, we're going to do an unencapsulated secondary lymphoid tissue. This is the Peyer's patch. And so in the Peyer's patch, you have a cell that filters things that are in your small intestine, and these are called M cells. And beneath these M cells is an aggregate of lymphoid tissue that's called the Peyer's patch. The Peyer's patch will have your antigen-presenting cells or your dendritic cells and macrophages right beneath those M cells to pick up whatever's coming through. You will have a B cell zone. You will then have in the center of that a germinal center. This is also called the follicle. And then you'll have your T cell zone. It's not encapsulated, so the cells are a little bit more free, but they tend to aggregate into their different zones based on the type and based on chemokines and cytokines that direct them to those sites. Okay, so that's the end of the first mini lecture, and I look forward to the second.